Hi, I'm Dr. Triparna Sen. I'm an assistant attending at Memorial Sloan Kettering Cancer Center in New York, United States. And I spoke at ESMO TAT conference regarding the novel biological characteristics and therapeutic targets for STK11 and KEAP1 co-mutant lung adenocarcinoma. So for a very long time, treatment decisions for lung adenocarcinoma were mainly based on driver mutation status. Until quite recently, we understood the importance of having co-alterations like loss of function mutation in STK11 and KEAP1. In our study and in some of the studies published in the past, we have seen that these co-alterations actually determine how resistant these cancers would be to therapies and how aggressive these cancers would be. In this particular study, we made several preclinical models in the lab and of either a single mutation in STK11 or KEAP1 or a co-alteration in STK11 and KEAP1. And we looked into the functional characteristics of these mutants, and we looked into the unique gene expression programs through bulk RNA sequencing. And of course, our ultimate goal is to identify novel therapeutic targets, for which we also did a CRISPR-Cas9 screen. Interestingly, what we found was that having a co-alteration in STK11 and KEAP1 actually led to an increased proliferative and invasive potential of lung adenocarcinoma cells. And not only that, these double mutants were actually turning on the ferroptosis protective genes. So ferroptosis is a non-apoptotic form of cell death, and these cells were actually evading or escaping this form of cell death. And we particularly found that one gene, SCD1, or steryl-CoA desaturase 1, was one of the factors that was significantly upregulated at the gene expression level in bulk RNA sequencing data. And not only that, CRISPR-Cas9 screen, we found that this was one of the, SCD1 was one of the genetic dependencies. What that means is that these co-mutant lung adenocarcinoma models actually needed SCD1 in order to survive. Now, when we either did a genetic knockdown of SCD1, we were able to completely kill these double mutant cells. And also pharmacological inhibition of SCD1 led to decrease in tumor growth in mice and also decrease in cell viability of these double mutant models. So sort of just summarizing, what we found was that STK11 and KEAP1 co-mutation actually predicts poor outcome for lung adenocarcinoma patients, especially metastatic lung adenocarcinoma patients. And these cells are extremely proliferative and they induce higher tumor growth. Now, these cells, as we have found in our study, actually turn on the ferroptosis evasion mechanism, which we believe is driven through the gene SCD1. And inhibition of SCD1 could be a novel therapeutic target for this subset of lung adenocarcinoma. Now, what does this mean clinically? We think that SCD1 is a good target for the STK11 and KEAP1 co-mutant lung adenocarcinoma. And we want to develop better targeted therapies for SCD1 because the current drugs that we have in the clinic are not that potent and not that specific. So next steps would be to develop specific and potent SCD1 inhibitor. And not only that, to test these regimens in the clinic along with existing chemotherapy, immunotherapy, and also with ferroptosis inducers that are currently in clinical trial. Because the ultimate goal of our group and for people doing research in lung adenocarcinoma is to develop personalized therapeutic options for these patients. And we believe by targeting SCD1, this could be achieved.